This section introduces the primitive the whole book is built on. Population genomics is population genetics, run to the whole genome limit. The object is the same, only N is larger. The chapter fixes that one primitive genotype variation across individuals in space and time, together with the forces that shape it. Population genetics studies this variation at a handful of loci. Genomics studies genome structure and function. Their product is population genetics with the locus count scaled toward the entire genetics with the locus count scaled toward the entire genome. What changes is N alone. The surprise that updates your model sits in the history. The discipline was born with Fisher in 1930, before anyone had proven that DNA carries heredity, which came with Avery in 1944. The abstraction was always locus first and substrate agnostic. Sequencing only supplied the loci. This section explains what population genomics is at its root and the three forces that generate everything it measures. Population genomics is population genetics, with the locus count sent to the whole genome. No new concept, only scale. The generator underneath is this: a population is a distribution over allele frequencies, and evolution is its stochastic dynamics. Three forces move that distribution. Mutation is the source, it manufactures the variance. Drift is the noise, random resampling each generation, with an expected fixation time of 4 times NE. Selection is the bias, fitness tilts the frequencies. Every later chapter estimates or infers one of these three from sequence data. The quiet surprise is how few primitives carry the field. Given allele frequency as a distribution, plus source, noise, and bias, you can predict what any population genomic statistic must be measuring. This chapter explains where the data comes from, the wet lab boundary that every later computation depends on. Everything computational in this book is downstream of one irreversible step: getting DNA out of an organism. This chapter fixes the input boundary of the pipeline, the single point where information enters and can only be lost thereafter, never created where information enters and can only be lost thereafter, never created. Its logic is conservation. A sample carries a finite quantity of signal set by physics before any algorithm runs. The mass formula, approximately 1.023 picograms per gigabase times ploidy, tells you how much DNA a cell holds. Ancient DNA a cell holds. Ancient DNA half-lives tell you how fast that signal decays. Sampling design decides which questions the eventual data can even answer. The generative move is this. Every later chapter inherits the envelope this stage defines. Computation redistributes information. The wet lab and the field are where it is won or forfeited. This section explains why genomic data acquisition is the first constraint on everything that follows. Genomic data never arrives raw. It is pre-shaped by the physical facts of its extraction. And this chapter's thesis is that acquisition is backward constraint propagation. The question fixes the marker method. The method fixes the tissue and DNA mass required, and the tissue fixes what decay and epigenetic state you inherit. The wonder is that the substrate is deterministic enough to compute before you sample. One eukaryotic cell is about 1 nanogram, so DNA yield is roughly genome size times ploidy times mass. Ancient DNA half-life falls predictably with temperature and fragment length. Every downstream analysis is thus prepaid in grams of tissue in degrees Celsius. Sampling design is the point where the whole book's inferences are silently budgeted. This part explains how every genomic object in R shares a single underlying shape. Every genomic object in R reduces to one shape, a flat vector of bits plus a pointer to attributes, the dimensions, the names, the levels, the class that tell generic functions how to read it. A class adds no new substance. It is a tag, routing the same bytes to the right method. So, the loci and genin encodings of one genotype set carry identical information, differing only in the shape convenient for a given analysis. Representation follows the intended computation. The dist example is the whole move in miniature. Symmetry and a zero diagonal collapse n squared distances into a vector of length, n times n minus 1 divided by 2, and the omitted structure is restored entirely by attributes. Learn the vector plus attributes. Primitive, and every genomic class becomes predictable. This section explains why, in R, every object reduces to one underlying thing. Everything in R is a vector. A type is just a vector wearing attributes, which are themselves vectors, and those attributes tell the interpreter how to read the same underlying bits. A factor is integers tagged with levels. A matrix is a vector tagged with a dimension attribute. A class string is nothing but a dispatch label. So the genomic classes this chapter builds, the loci class and the dist anchor that folds a symmetric distance matrix down into a vector of length n times n minus 1 divided by 2 plus a handful of attributes introduce no new kind of thing. Structure lives in the tags, not in the substrate. The chapter's role in the book is to establish that every downstream genomic object is a base vector plus an interpretation, so that later analyses compose over one atom. This section explains how a single R operator covers everything you will ever do to data. 3 operations: exhaust data, manipulation, subset, replace, and delete. R expresses all 3 through one generic operator, the square bracket. The chapter's move is that this operator does not care what it indexes. Real numbers, integers, DNA bases, genotypes, alleles all answer the same syntax because each class registers a method behind the one interface. Learn the operator once, and every genomic type inherits it. This is dispatch as compression. The behavior lives in the operator. The data types merely plug in, so competence transfers instead of multiplying. The 2 object systems fit the same grammar. S3 objects, such as loci and D and abin, wear a class attribute over ordinary vectors. S4 objects expose their slots through the @ sign. Both bow to the same indexing grammar. The chapter is the toolbox that the later case study chapters will reach into. This part explains what the 4th structural primitive of the chapter does, why one operator carries the whole design. One operator does everything. R's bracket operator is generic, dispatched by class, so subsetting, replacement, and deletion read identically whether the elements are integers, bases, alleles, or genotypes. A data class is thereby what it does under the operators, not what it stores. Silent coercion and the as-dot conversion morphisms close the loop. Chapter 3, Zoo of Classes, Loci, DN, Abin, Genind, and X, string becomes a connected graph you traverse at will. This runs over a copy on modify memory model that makes the assignment Y gets X free until one of them is touched. This representation invariance is the chapter's work. It turns a catalog of types into one uniform substrate. The later analyses run on, and seats the case study datasets there. This section explains why every summary method in the chapter is a way of compressing before you fit anything. Before you fit anything, you compress. Every method in the chapter is one lossy projection of the raw genotype matrix onto a facet, cheap enough to see, an allele frequency, a diversity index, a pairwise distance, a variance maximizing axis. Exploration is choosing which projection exposes the structure, the group differences, the structure, the group differences, the missingness, the relatedness that you will later think. The sections form a ladder of increasing aggression. First counting alleles, then per site diversity, then the full pairwise distance matrix terminating at PCA, where the whole matrix collapses onto its dominant eigenvectors. Distance and PCA are the two poles. They are the same information read pairwise versus decomposed. Pick by whether the structure lives between samples or across them. This section explains the single move underneath every tool in the chapter. Before any model or test, there is one move. Collapse the genotype matrix along a chosen axis and read what survives. Every tool in the chapter is that same map, a large table sent to a small legible object, differing only in which axis you pick. Per locus yields allele frequencies, per site nucleotide diversity, per pair genetic distances, per group stratified summaries, per position window a smooth genomic profile. The axis of collapse is the entire design space. Ordination, meaning PCA and MDS, is the limit case. Here, the data names its own axis, the direction of maximal variance, instead of you imposing one. This stage precedes inference because the projection you choose is what renders structure visible at all. The hypothesis comes after you have seen it. This section explains linkage, disequilibrium, and haplotype structure, and the single move that runs through the whole chapter. Two loci sit in linkage disequilibrium when the haplotypes you observe deviate from the ones allele frequencies alone would predict. The quantity d sub a, b, defined as the probability of the AB haplotype minus p sub a times p sub b, is nothing but the covariance of two alleles, and r is its correlation. So the whole chapter is one move, a munus. This is the same chi squared against a contingency table structure you already own. Now read across chromosomes. Complete linkage disequilibrium is the ceiling. Take the toy 2 locus example, locus 1 and locus 2, where allele A rides only ever with allele C. The observed haplotypes collapse to the diagonal, and R equals 1. Recombination is the eraser that pulls R back towards 0 with distance. Haplotype blocks are simply the regions where it hasn't finished erasing. This section explains linkage disequilibrium as a single statistical primitive. Two loci sitting on the same chromosome betray that fact statistically. The gametes they co-occur in appear more often than chance would mint them. That is the whole primitive. Linkage disequilibrium is an independence test. Read as a map. Start by predicting haplotype frequencies as the product of allele frequencies, PA times PB, which is the null hypothesis of no association. The gap between predicted and observed is the disequilibrium itself. Written out, D between allele A and allele B equals the observed frequency of the AB haplotype minus PA times PB. Its normalized form, the OB haplotype minus PA times PB. Its normalized form, the quantity R relation coefficient. Recombination is the decorrelating force. Distance along the chromosome erodes D toward 0. So physical structure, meaning which loci travel together, falls out of a chi-squared flavored frequency mismatch, never needing to see the chromosome directly. The chapter is a catalog of R functions computing this one contrast. This section explains population genetic structure as departure from random mating. Structure is nothing but departure from panmixia. Hardy and Weinberg fix the null. Random mating turns allele frequencies into genotype frequencies p squared, 2pq, and q squared. Every genotype excess or deficit against that null is structure, made visible. Wright's own epigraph sets the chapter's problem. One scalar of f cannot hold it, so partition the heterozygosity deficit by level, individuals within a dme, written FIS, and dmes within the whole, written FST. The levels are nested and multiplicative. 1 minus FIT equals 1 minus FST times 1 minus FIT equals 1 minus FIT equals 1 minus FST. The chapter's whole arc is the enrichment of that single number once it proves too coarse, networks, ordination, Bayesian clustering, and admixture. Every method reads one signal. Where does reality diverge from random mating, and at which stratum does the divergence live? This section explains how population structure reduces to a single comparison, reapplied at every level of nesting. Structure is a deficit of heterozygotes against what random mating would produce. That single comparison, reapplied at each level of nesting, generates the whole chapter. Hardy-Weinberg fixes the null. Under panmixia, it predicts the genotype frequencies p squared, 2p, and q squared from allele frequencies alone. Every departure from that is structure. Wright's epigraph 1f is never enough is the thesis. You measure it as 1 minus the observed heterozygosity divided by the expected heterozygosity, choosing which two heterozygosities fill the ratio. Individuals within a subpopulation give FIS. Subpopulations within the total give FST. Individuals against the total give FIT. It is one shape indexed by which nesting level you compare. Know the ratio, and you regenerate all three plus their chain. 1 minus FIT equals 1 minus FST times 1 minus FIS. This section explains how geography enters population genetics. It enters through a single substitution, the weight matrix. Variance is mean squared distance between individuals, and Escoffier's AMOVA rests on exactly that equivalence. So every structure statistic is one quadratic form, X transpose times W times X, and W is where space lives. Ordinary PCA sets W equal to the identity matrix, treating everyone as equidistant. SPCA swaps in a neighborhood matrix, computing covariance only among spatial neighbors. Moran's I weights each allele, frequency pair by geographic proximity, and AMOVA's nested hierarchy. The coefficients phi CT and phi SC is merely a block-structured PHE, and phi SCC is merely a block-structured W. Here is the surprise the chapter understates. Choosing how proximity is measured, whether Euclidean, geodesic, or landscape cost distance, that choice is the modeling decision. The algebra underneath never moves. This section explains how geography enters population genetics. Geography adds no new statistic. It reweights an old one. The chapter's single move is this: population structure is total genetic variance partitioned across nested levels, with continent containing population containing individual. AMOVA is that partition, and the phi statistics are its ratios. Each phi is the fraction of variance a given level explains, the exact analog of FST. Space enters only by softening the categorical level into a proximity. Moran's I weights pairs by distance instead of by shared label. Spatial PCA literally swaps the n by n identity matrix inside ordinary PCA for a neighborhood matrix W, so covariances count only near individuals. Where is just which group made continuous? One decomposition, two ways to name the neighbors. This part explains how present genetic diversity records the size a population had in the past and how one move reads that history back out. Present genetic diversity is a photograph of past population size, developed backward. The coalescent runs genealogy in reverse. It traces the sampled lineages toward their ancestors, and each merge fires at a rate proportional to 1/n. A large population gives rare merges and a deep tree. A small population gives rare merges and a deep tree. So the sequence of coalescence intervals is itself the size history, n of t. A growing population leaves short waiting times near the present. A shrinking one leaves long ones. Kingman's exponential intervals carry the whole signal. Every past demographic event is recovered by one move, inversion. The shape of the gene tree encodes the demography that generated it, and reading population history off present-day sequences means undoing that map. This section explains why reading ancestry backwards turns an intractable problem into a trivial one. Read ancestry backwards and the intractable becomes trivial. Forward simulation tracks every one of the n individuals reproducing. The coalescent tracks only your n-sampled lineages merging into shared ancestors, so cost scales with the sample, not the population. Here is the primitive. Two lineages coalesce each generation with probability 1/n. So waiting times are exponential with rate set by n. This means the distribution of merge times is a direct readout of past population size. A growing population compresses coalescent times toward the present. A shrinking one stretches coalescent times toward the present. Genetic diversity theta equal to 2 times n times mu falls straight out of expected tree length. So the chapter's whole inference engine reduces to one move. The shape of the coalescent tree is the demographic history, and estimating branch lengths estimates the past. This section explains natural selection as a structural primitive. Selection is never observed directly. It is inferred as the residual left over after neutrality is subtracted. That single move is the whole chapter as one primitive. Every method builds a quantity that must take a known value if only drift and must take a known value if only drift and mutation are acting. Consider two of these methods. Tajima's D subtracts two estimators of the same population parameter. Theta, two estimators that agree only under neutrality. The ratio dn/ds compares the non-synonymous substitution rate to the synonymous substitution rate, where equality means no pressure at the protein level, and the ratio's departure from one localizes that pressure. The move is identical across all of them. Construct a null prediction, then measure the divergence from it. Selection is a departure detector, and that is why every test here is a neutrality test. This section explains how population geneticists detect natural selection, and the single move every test in the chapter shares. Selection is never observed directly. It is the residual left after neutrality is subtracted. Every test in the chapter is one move. Build what drift and neutral mutation alone predict, then measure the gap. Tajima's D pits two estimators of theta that must coincide under neutrality. The dn to ds ratio asks whether protein-changing substitutions outpace silent ones. FST outlier scans exploit that drift strikes every locus equally, so a locus differentiating too much betrays a force acting on it alone. Extended haplotype homozygosity catches a force acting on it alone. Extended haplotype it drags behind. One null. Many observables. Selection is wherever the observable refuses to match the null.